So, behavioral and cognitive studies suggest that children develop more complex conceptions of emotion and expressions of emotion across normative development and into adulthood. Between these periods, there are a lot of changes in the connectivity and structure of regions, which might exhibit neuromodulatory effects relevant to emotion. It may be the case that as these structures develop, how individuals represent emotions or the ways in which they mentally render characteristics of the concept become more complex as well. And this is relevant because representations may have an influence on downstream processes that affect future cognition and behavior. So, to determine whether or not these differences in affective representation exist, we had asked 36 children and 21 adults to passively watch a custom stimuli set while undergoing an fMRI scan. One adult and 11 children were excluded from the final data set for excessive head motion. And the stimuli set contained 8 positive and 8 negative clips from 8 popular children's movies, such that each movie provided both a positive and negative clip. Clips contained both animated and live action movies, anthropomorphic and human characters, and span a long time period with release dates from 1939 to 2011. In terms of quality control, no group differences in familiarity were observed between adults and children, and independent raters counterbalanced clips on a second-by-second basis for affective intensity, affective presence, and the presence of social stimuli. We'd identified 3 regions relevant to our question: the amygdala, the nucleus accumbens within the ventral striatum, and then the ventromedial prefrontal cortex. And we chose these sites because the literature suggests that all 3 should represent affective information and may undergo these structural and functional changes over normative development. From each of our 3 regions, we'd captured patterns of changes in neural activation over subsequent stimuli presentations. And by correlating neural activation patterns across stimuli of the same valence and within the same ROI, we can use a method called representational similarity analysis to arrive at a value that symbolizes the strength of the representation of that valence for a particular region. So in other words, when a region that produces a highly similar pattern in response to different stimuli holding one characteristic in common, it can be inferred that the observed response is a product of that common characteristic. Once we arrive at those values, which are essentially just nonparametric correlations, one could spatially analyze the correlation matrix they'd produced or refer to a more traditional analysis of variance. So with that in mind, we hypothesized that adults would generate less pattern similarity than children. And this is based on the theory that they'll entertain more complex conceptions of emotion. We also hypothesized that children would generate a greater difference between positive and negative pattern similarity than adults. And lastly, because of the profound developmental changes associated with the VMPFC relative to subcortical structures, we hypothesized the interaction between subcortical structures and the VMPFC would produce greater variance in representational pattern similarity for children relative to adults. After computing our RSA values using a Spearman rank correlation and applying a Fisher Z transformation, our values were entered into a 3x2x2 mixed effects ANOVA with terms for ROI, age group, valence, and interactions among them. Follow-up contrasts were Bonferroni adjusted, and supplemental to our primary analysis, we explored the coherence of our child grouping. We did this by entering age as a predictor towards pattern similarity in a mixed effects linear regression model, and we found no relationship among children by age, suggesting no reason for us to treat the values we observed from a child at age 4 any differently than we would from a child at age 10. In all cases, we supported our primary analyses, our primary hypotheses. Children did demonstrate greater pattern similarity relative to adults. Children also demonstrated greater negative pattern similarity relative to positive pattern similarity, an effect which was not mirrored in their older counterparts. And then lastly, adults demonstrated no ROI differences, while children demonstrated differences between the VMPFC and each subcortical structure. All results are also captured in the supplemental materials. All in all, these results do suggest that differences in affective representation exist between adults and children. Being put in the context of the extant research, this may reflect a maturation from visceral emotional responses to more evaluative processes. However, given these analyses, we cannot speak with any certainty as to exactly what about these representations are similar, what mechanisms might contribute to a change in representation, and whether these representations do have any meaningful impact on behavior, and future analyses really are needed to explore those relationships.